How's your
What?
Cartier model?
I'm sorry?
How was your Cartier model?
Oh, he's outside chasing little lizards.
Fabulous. Did you know his cat has a Cartier, like a collar?
A
No,
Cartier?
yeah, the pendant is Cartier and the collar is what do you call it?
Is it Tiffany or something?
Yeah, Tiffany's.
See, I remember it because I have none. That's great. That's awesome. That's awesome.
That's so
Great.
funny. Mine is
He's
right
a
here.
he's a pretentious little cat. Oh, there he is.
Wow. That's a chubby one. He's cute.
She's gotten chubby in the last little bit. We just switched our food to like something that's more fish based, and she's been smashing it. And the
Oh,
other cat,
wow.
we have two. The other cat has literally pretty much just been eating the dog food. Like she will come when it's time for the dog to get fed in the evening, especially. She'll come and like bump up against me and get all underfoot and try and get me to give her the dog food first. And she will go and eat the dog's food. And we have a 80-pound German Shepherd, and they're like 10-pound cats. She just goes and bumps the dog out of the way and eats her fill first, and then Athena, the dog, goes and eats her food.
See? Yeah. That's awesome.
So
Yeah.
funny.
I'm waiting for my kids to be out of the house so that I can have my four-legged babies. Not yet. So
You
Fair.
don't have any
I
pets,
cannot.
Padmini?
Sorry?
You don't have any pets?
No. No. I'm waiting. I'm waiting for my kids to go to college so that I can have my four-legged babies.
Okay,
So yeah.
cat or
My
dog?
you know, it might be controversial. I am kind of a dog person because I used to have a cat and I loved it so much, but she was so selfish. She did not give any love back to me, and she always brought me back trouble. So it's like now I'm thinking maybe I should have a dog. I had both. I grew up with both dog and a cat. So maybe I will like y'all's cat look really great. Maybe I'll just fold and get a cat also. As of
If
now,
you
I'm thinking of a big dog.
if you want something cuddly, get a Munchkin cat like mine. Munchkins
Yeah,
are
those are
very cuddly.
yeah. I want
Really.
something that needs me because I need some attention. So
You can't go wrong with a dog then. Cats,
yeah, right.
Oh,
That's why
it's
I'm
such
like,
a crapshoot with a cat.
yeah, I always need a bigger dog too because I do not like I don't know if you've seen me in person, but I'm tall. And
yeah.
I feel like anything smaller, I might just step on them, which I really don't want to. I love big dogs. So I'm thinking, yeah. I have my fixes, but I live in Texas, so I have to be mindful of that too.
Oh yeah, you
If
Mm.
cannot get like a husky because then it's
Oh,
so
I've seen
sad.
huskies, but they are maintenance. You have to feed them like really, really cold food because it's really hot here, and you have to have the temperature of the house at And I'm an Asian household. You do not touch the thermostat at all. So I cannot have a husky. Yeah, if I had my way, I would have a Saint Bernard, but I'm not
Oh my
in
God.
UK. I would
That's
love to have a
big
Saint
dog.
Bernard. So that big of a dog is what I'm looking for. So let's see.
Maybe a Great Dane or something. I feel like they're a little shorter hair.
Yeah, the Great Dane will be an awesome dog, and we have like similar dogs in India. If I had my choice, I would try to bring an Indian breed here, but I don't know whether we can bring in Indian breeds. So
You
that's
can as long as that they're quarantined and
hmm.
they need all the vet shots and a declaration because that's how I
Uh-huh.
brought my cat into the U.S.
Oh, okay. Awesome. So, like, if it's my dream dog would be a breed called as Rajapalayam, that is literally a South Indian breed of dog, which is not found anywhere outside. So, if I'm able to bring it in, that'll be awesome. Let's see.
Later on, send me a picture of it because I've never heard of this dog and I don't know how to
Oh,
type it.
oh,
I was
yeah,
just
yeah,
trying
yeah.
to
I
look
will
it up, but
definitely
I
send
don't
it
know
to
how
you.
to.
Those are majestic dogs. Those were companions for kings in India.
Oh,
So, those are like hunting dogs, though. So, really great dogs.
yeah, I can't say the same about my people's dog. The dog that used to hang out with the Aztec nobles is
Uh-huh.
so ugly.
Duh.
Yeah,
It looks
it
like
looks
something
so
that crawled out of hell to give people nightmares. So
You're going up to get sacrificed, and you see a pack of these dogs standing on top of the pyramid, and you're just like out of
That's
it.
yeah, that's pretty much it. And the problem with those dogs too is that they're they're highly intelligent. They're actually considered one of the most intelligent breeds, but they're not very loyal. Like if they feel that you're
That's why
not
they're
treating them
intelligent.
well, that's the thing that
Intelligent people, I can tell you, are not that lost.
no, like they're very loyal if they feel you're treating them right. But if they feel you're shortchanging them, they're known to just like walk out of the household and just be like,
Yeah, it
"Bye."
knows its worth. That's why it's intelligent. Oh my God, I would love to have the breed. It knows its worth. Come on now. It's like my daughter. She will walk out of here if she feels like you are not treating her right, which is that great girl. You do that. So yeah, I would love for to have a dog like that.
No, but have you? It's a really ugly dog.
What is it
Oh,
called?
A Sochelt.
now I am super
So
curious.
cool.
Eric, if you
Yeah.
find it, share it with me.
Let me see.
My spelling is real bad this morning. Oh, XOXOCHITL.
Mhm.
Now I'm super curious to see it. Oh my God, my Rajapalyan looks similar to that only. Oh yeah, I see that. I see that. But
Not that ugly.
it's cute.
Look at
Human
this.
being,
Look at
it's
this thing.
hideous. Look at the one in the third picture. Like they could be really pretty, like the one that's on the Reddit. But I think
Oh,
that, yeah, I think that one
That's
was
an
probably
exceptional.
photoshopped.
that's like
Yeah.
wearing a mask, but I would love
Yeah,
to have it.
that's like the real one, like the where you're putting it at. That's what it actually looks like.
It looks kind of like a mouse.
Doesn't
Yeah.
it?
Mousy looking. Yeah.
Well, I like the one in the Reddit. That looks menacing.
Yeah, that's a cool looking dog.
Yeah,
That is an awesome looking dog.
I don't know. I think that one is photoshopped.
That's like the Hounds of Hell kind of looking dog. That makes perfect sense.
Yeah,
Yeah, I think
but
it's pretty
but
cool looking.
but look at the other one, like the one right next to that with the little
This one,
white streak on its head, and the one right below that.
But still, though, it can be cute and looking.
this is a
Oh,
funny picture all around.
you want? Then you should definitely look at my dog. Let me share.
Yes,
I don't know how
show
to
us
do
What's
this thing.
what's
spelling.
I'm gonna drop this thing in the chat, and Eric might just open it.
yeah, we're recording by the way, just so we can have all this because this is pretty good stuff.
Yep, definitely. This is what we are. So this is what I would love to have if I have my choice.
This is what scientists do on Fridays.
I dropped the link the in the chat. But you know what? I wouldn't mind this dog.
Really?
Yeah. I'll actually like yeah,
That's a
these are the
great
ones
dog.
I'm looking for.
Oh yeah,
It
that's
looks
a
kind of
really
like
pretty dog.
almost like a mix of a lab and a Great Dane.
Yeah,
Hmm.
right? So they have a really high arch. So they are hunting dogs and they are like they are bred for like the races and stuff. So it's really cool looking dog.
Yeah.
And
How
it's
friendly
mainly
are they?
because sorry?
Are they really friendly?
Yes, they are. And they are awesome watchdogs. But thing is, you don't get the purest bred. It's really hard to find one these
Mm.
days. So it's really hard. But yeah, they are like a hound,
Yeah, he looks really pretty.
right? So
Mhm.
this is looking pretty because it's white, but it comes in different colors. So it's really great. The browns are fantastic. Yeah. It's
Oh, this
hard to
looks
find
cool.
purebreds right now. See, yeah. See that brown one is fabulous looking. But
Yeah.
yeah, I would love to have this one. If I had a choice, that's my dog.
You need to bring it from back home, Padmini.
Yes, yes. I'm going to start floating the word maybe in a couple of years because my kids will be starting to move out of my house and then I'm getting my baby. Because I want to do good by all the people around me. So I want to be able to give my attention to all of them. then I'll be able to do this.
That's
Yeah.
true.
That is
Pets do
fair.
require a lot of
Yeah,
attention.
I see. Definitely, my attention has gotten split between all the pets.
Right?
Yeah. Here's our pup.
Oh my
Oh, yeah.
God. Oh my God. So beautiful. Oh my God. Yes.
Yeah,
She's,
she's a sweetheart.
yeah.
She does look like super friendly.
She is so friendly. Yeah, she was a police dog reject because she was too
Too
sweet
big.
and too anxious. So we got her. And it checks out. She has a big bark, but she's such a softie.
Yeah. Great.
Oh,
That's
Funny.
yeah.
awesome. That's awesome. As of now, I just have some fishes. That's it. Yeah.
Fishes or
That's
fish?
what cat.
Sorry?
Or
Like can't get a
fish?
cat.
Oh,
I don't have a cat, but I would love to. But I have fishes as of now. I
you have a
used to have
clownfish?
a 50-gallon tank. I
Wow.
have downgraded to a 50 because I don't have time to maintain them. So,
There's this thing that just came out in Japan that does the automated aquarium cleaning. I just saw it on TV the other day.
maybe.
Like you put it inside the aquarium and it sucks up all the debris and the
Mhm.
water just continues like spilling out. So it has like a mesh inside, but it sucks up everything and it
And then
cleans
it is
under the
there.
pebbles.
Oh, okay. That's nice. Nice to know. Then maybe I should just dust off my 50-gallon one. Yeah, we used to have a really great aquarium, but we had this Harvey happen, and then we had to like leave our house for a week. And then, oh my God, that was a mess. And then I lost my fishes. So I was like, okay, fine.
Mm.
I'll come back to this later.
That's sad.
It is, it is. But it's awesome to have them because gives you like makes you feel calm just sitting there and looking at them. And my husband loves to do that too. So we kind of had it as a hobby. Maybe we should bring it back.
Good idea.
Definitely. Okay. If you guys don't mind if we switch gears a little bit to flow.
Yes, sir.
Sure.
Definitely.
Yeah.
The recent
You want
uh,
to introduce Padmini for our audience who we kind of just jumped into things talking about kids and pets and everything, but maybe let's take a step back and welcome Padmini to our Go with the Flow podcast.
Yeah, I literally went with the flow, didn't I?
That's what it's all about. I love it.
Yeah, yeah, awesome. So that's what I'm known for, going with the flow, actually. So I am Padmini. If you know FlouroFinder, you heard my name there also. I work with FlouroFinder, and most of the emails comes out saying, Padmini from FlouroFinder says this. That's me. So I work there as, I started as a sales manager there, but now I am the partnership manager and community growth. So I kind of try to go talk to people about FlouroFinder and you tell them why you have to use it or why you can use it. So that's what I do. But I am a flow cytometrist in heart. Been working with flow cytometry since my grad days in Cleveland State, Cleveland Clinic, and then in Baylor as a postdoc for 5 years, and then worked in the flow core for 3 years. So for more than 15 years, I've been just doing flow or associated with flow cytometry. So I just made it as a career right now, I feel. So yeah, I'm really happy to be here, Christian and Eric. Thank you for bringing me in.
we're really
For
happy you were
sure.
able to join
Yeah,
us.
Yeah, dude,
absolutely.
it's awesome.
Yeah, I'm curious because I didn't know you worked in a flow core. I also worked in a core. And I'm curious, like, how that jump from the core to industry went, and kind of why you decided to do that.
For me, I knew it's like the jump from postdoc to core facility was something that happened serendipitously. Like I was not even applying to the core facilities, but I used to use them a lot and I was looking and I knew that I'm not going to be writing any more grants. So it's like they asked me, "Hey, do you want to join us?" Like, yeah, I would definitely join you. So that was a really unfortunate thing that happened to me. But the switch from the flow core to industry was a deliberate choice because it was mainly a personal choice, nothing to do with the flow cytometry core facility in itself, because I loved doing what I was doing. I used to train the users and I used to do high parameter panel designs and stuff. So it was fantastic because I get to do the science and not care about the science at the same time. So I was really great. So you need to be there at this set time, and then it was a rigid schedule. That was the main thing that I had to change a little bit because my kids were going into middle school, and I had to be around and be a chauffeur for them. So I needed to be able to pick up and drop off and go wherever I needed to. That's why I wanted to do something in an industry, or not just industry, but anything that gave me the flexibility of schedule. That's what I found in industry. So that's the main jump. So I actually tried to become a technical like associate like TAS, but I did not get that. Like I joined plus Slingshot Biosciences, and they asked me to be a technical sales specialist than an FAS. So that's how I started my career in sales or in industry. So that was a choice because of the timing, nothing to do with the core facility. If I had my choices, I would have definitely stayed in the core facility for a longer time. But the money doesn't hurt either. So
Interesting.
So I have a question for you, Padmini. See, you were doing it sound from your description at the core, you
uh-huh.
were doing high dimensional panel design
Yeah.
or panel design for high dimensional flow. I think that's
Uh-huh.
more accurately phrased. How have you seen panel design change since your core days to the present date?
I can tell you, and I cannot believe this is just like 4 years into where I'm right now. So 4 years ago, high parameter was taught, or at least where I was doing 20 plus colors, we considered as high parameter. Now I feel like with the explosion of what is it, full spectral photometry, 20 plus is a regular mundane thing. Everybody does that, right? Now it's all like, how can I multiplex it? How can I bring in the like put together two different panels to make a bigger panel? And that is considered like anything that 30 plus is what is considered as multiplexing or high parameter right now. But I feel like people are much more aware of controls these days, a little bit at least. I feel like because when I was in the core facility, nothing against the people who I was working with, but we had to tell them, hey, you need a control for every experiment. Now I feel like people understand that because, yeah, and I'm thinking this is more because with the growth of the spectral photometry, because it's not just one instrument. Now every instrument company has a spectral flow, and you are moving into that era where it is a necessity to know about that. And that with that comes the understanding of autofluorescence, kind of understanding of controls and all those things. I feel like much more awareness is there in the hyperameter space right now, and that is very encouraging for me. And even when we went to all these core the user group meetings and all these conferences that we go to, we have moved away from I don't want to say we are moving away from the actual like research, but we are talking more about the how do you create a better panel for this? How do you create a reproducible panel? What are like those kind of talks are much more
Quality.
prominent, yeah, right now than somebody comes and talks to them about, hey, this is the paper, and I mean, this is an research project I'm working on. Like it's much more generalized. It's not like very niched right now. People are more talking about all these rigor and reproducibility controls and all those things. I feel like we are in high parameter panels have become much more common. Like even when you're doing an exploratory research, you're still wanting to do a bigger panel rather than say, hey, I'm going to sit with just smaller panel. Like now, everybody wants you to bigger panel. And I feel like that awareness has come through, and that's what is making them much more conducive to start running these. So yeah, I'm very happy about where we are going for sure. High parameter.
I'm I'm curious on the controls. Do you see that as like, do you see that across the board of people being more aware of the need for controls, partially because of the ubiquity of hyperparameter, or do you think it might be influenced somewhat by like working for FlouroFinder, where people are going to go and seek out these resources and knowledge uh, repositories, basically, to help them perform better experiments? Do you think that that's kind of a self-selecting group that are more worried about the quality, and that the average user is probably like in a core, like the same thing? I've experienced that too, of where people are like, "Oh, I need controls. What do you mean I need controls?" So, do you think there's still some of that in the average
Yeah, I
case?
feel like I'm thinking this is because the new generation of PIs or postdocs who are training these newer users or someone we have worked with. We have harped on saying, "Hey, you need controls," and they are they are understanding that. I mean, they understood that, and then they are able to impart that knowledge to the ones that are coming in. And why I'm able to say that is because of the request that we get in FlouroFinder from our users saying, "Hey, can you put us more information about controls?" That literally brought us to set up create a feature in our panel builder where you actually have a space where you can go and select controls. So we show you what are the controls that are available for you to, because people are expecting to see that. That means there's a basic knowledge within the community where they know that, okay, fine, I need controls, right?
That's
Which
really
is
good to hear.
that is fantastic. I'm very, very happy to see that because apart from doing all these trade shows and talking to like doing sales, I also do quality control and the product development on FlouroFinder. And I was super happy when we got that request, and I was able to go in and create that module or help them create that module. So it's like the core facility instructor within me was very, very happy on that day, thinking, oh my God, people are thinking about controls, right? So yeah, and I feel like that it's because of what we all did, you and me, Eric, like core facility managers and core facility staff trying to impart like, hey, you need controls. Like you can't just willy-nilly come and do a gating strategy. Like you definitely need an unstained. Please bring me that, right? So I feel like there is a general understanding of people on, hey, I need to have all these things to actually create better data, right? I just don't want to come and just run sales stuff. And I'm also thinking it's because reagents are becoming a little bit pricier, right? Let's not run. And I don't want to get political here. We don't have a whole lot of money to just slosh around. And I'm thinking we are starting to plan our experiments better before we jump into doing something. I hope that's the
That's
hope.
music to my ears. That's what I try and do with people is like, look, if you plan it out well, you design it well, you plan it, you test it, it's going to work a lot better, and you're not going to end up wasting all your time and money with data that just isn't really usable for much. So
Right?
it
I totally loved it because I want to be very honest, Christian. I haven't read the entire paper yet. The 22 pages, but the ones that I got from your thing was you talking about, hey, think about everything before you even touch an instrument, right?
yeah, I
That's
like
something
that.
I loved.
Awesome.
Yeah, and in your paper too, Eric, the last paragraph when you're discussing about the experimental design and the conclusion of your discussion part, you talked about, hey, this is a very limited sampling that we had in there, which is again something people forget. Hey, I'm going to read a paper and they're talking to me about this thing, and this is something that is happening, right? No, you have to be able to say, hey, this is a limited sampling. This is a possible thing that could be happening, but we would like to have more information. So I felt like those two aspects are really, really important. It's not like you think about everything before you touch something. And also at the end, when you have a data and you have analyzed the data, think about whether it is actually universally applicable, right? So I love that thing about this, like the completion of the paper. I mean, I call it as a journey of the data. Like completion of the journey of the data was
Exactly.
fantastic there, right?
I
Yeah.
like that. I like that phrase, the journey of the data into well, I guess it starts as information, right? That's the journey. Information turning into data, turning into interpretable results. And you could only do those changes, or you can only get that evolution of the information if you plan it out correctly, because trash in, trash out. You're not going to get interpretable results if you don't plan out your experiments. And I know that it's the knee-jerk reaction. Well, I guess let me ask that from you guys, since both of you worked at a core, from like, let's say grad students, or I would say mostly from grad students, probably entry-level postdocs, that they get an experiment and they just go to the core thinking the core is going to do all the magic, but they themselves don't sit and think about, I need to do all these things before I put together my panel and run my actual assay, right? Because there's a lot of stuff in between.
Oh yeah.
And I just don't. I don't know. Back in the day when I was in academia, I saw that a lot. When I was in FloJo, I heard a lot of complaints from court directors that despite them having the resources for people to look them up, it was a lot of here's my data or here's my samples, and then there's no controls or there's controls missing, and or you did the acquisition and then they complain to the core, hey, where all my information? And then the core's like, well, you didn't acquire correctly, so that's gone. And then they get upset, right? So what's your take on this, and how do you think for those that are in SRLs, how do you guys think that now that you've been in the core, have transitioned out of the core, motivate people to start thinking about all the pre-planning that you just said how valuable it is, but I don't think you get that into your later stages of your career or later stages of your experience with Flow. But in the beginning, because I also did that in the beginning too, I just wanted to throw things in there and go get data, right? So what's your advice for people in SRLs or to those researchers that are still having those type of behaviors?
I mean, I think just like what Padmini brought up about the cost of things, I think something that gets forgotten a lot of times, especially in academia of late, like maybe more recently, this has become more brought more to the awareness of like, oh, these things actually cost something. Like if I use a whole box of pipette tips, like that's going to cost money to the lab that we might not have. And so like thinking more efficiently in terms of how you're running your experiments has to come into play if you have a limited budget. So then that leads to like kind of what we hit on earlier of, well, if I actually plan out this experiment first before I just willy-nilly just go and throw some antibodies in a tube and try and see what happens and hope for the best. Like let's take a more solid approach. Like it doesn't cost anything to sit down with your lab notebook and the core director or whoever and talk to them and say, this is what I have in mind. What do you think I can do with this? And another thing too that like I saw for people that are not familiar with flow is like some people just come and say, I just want to look at my samples and just give me the proteins that I'll see. Flow is not like sequencing where you can just like sequence everything and look at everything at once. Like you need to know what you want to look at. Right? Like that's a big
Mhm.
thing. And knowing that in advance helps you design the assay better, helps you qualify it, use whatever controls that you need. Like that sort of stuff, like sitting down and planning it, like we talked about, it just makes it much more efficient. And that's something where maybe SRL staff could start to help coach their users a little more because again, I don't think it really gets talked about a lot. It's the cost of doing things, period, and then the cost of doing something poorly versus doing it the right way and how like what that weighs. Like cost to usable data is really the like ratio to kind of figure out. And that's ultimately what matters. Like how much you're going to pay for usable data.
Mhm.
Yeah, I
Okay.
absolutely agree. And I feel like when it comes to SRLs and CDMOs and CROs, I feel like it's a much more important than I wouldn't say it's not important for academics. It's definitely important everywhere, but I feel like it's much more detrimental when it comes to SRLs and the CDMOs to be able to have this laid out plan saying, hey, this is how I'm going to do. And even when I was in the core facility, we used to have hours called as consultation hours. We never just accepted, hey, you can come and run a panel or something like that. We always
Mhm.
spoke with the user before we did anything. We had this hour and explained them, hey, what's going on? And they had to explain to us like what they need from us as well. So that's something we did. And I don't want it to sound like a sales pitch, but that's what FlouroFinder does, right? The main important reason why we lack in planning is because everything is everywhere. So not the information is all scattered around and you have to literally make an effort to find anything that you are looking for. But with FlouroFinder or platforms like I don't want to say other platforms, like definitely FlouroFinder actually has tools that brings everything together, because even when I'm speaking to anybody about FlouroFinder, what it does, it's actually has tools that can walk you from the start to finish, right? It's not just I tell them it's not just panel designing. You're not just designing a panel. You need to think about the biology first. Before you think about the biology, you need to understand what the question you're asking. So it
Mm.
makes you think about everything before you even touch a flow cytometer to run your assay, right? So you go to we have something called a cell ontology tool or cell types tool. You hop on that and you actually can find all the reagents that you require to narrow down to the population. It gives you gating strategy, making you think about your analysis even before you create a panel, right? So you need to be able to get on a platform or like find all the information that you need in one place so that you can confidently create a panel and be what is it, like you can be confident in saying, hey, whatever I'm running, the data from this is going to be better. I'm going to be able to interpret it better, and I'm going to trust the result that comes out of this, right? That trust and confidence comes only when you create an experiment better, right? So it's
Right.
not just, you can't just think I'm creating a panel. You're creating a panel that is going to be producing data. That data is going to be interpreted, or sorry, you're going to use analysis to analyze the data and get analysis, I mean, the data, your interpretation out of it. So it's a continuous process, not split into one or the other. So I feel like people need to understand that. Like they literally take an hour, sit down, think about it, have a powwow session, or have like a platform like this, hop on this and try to find something, whatever you're looking for, come up with a game plan. Other thing that you can do is literally reach out to us, right? Hey, because behind the screen, there are people who have worked with flow cytometry for a long time, are able to answer the questions or even connect with people who will be able to answer these questions. So always try to find these resources like this, connect with people, and then be confident in what you're doing, and then you can do whatever, right? Instead of just trying to put everything together in the kitchen sink and then hopefully you can publish the data. So let's not do that,
That's
right?
that's music to my ears on a Friday morning, honestly. That's exactly how I see it too, of like it's not separate things. It's all
No.
continuous step. And in my mind, honestly, the actual running on the instrument, that should be almost like an afterthought, sort of. Obviously, we all know there's like certain considerations you need to put in place in that, and you have to be precise in what you do. But really, it takes care of itself if everything else is well designed and well thought out. The running is the easy part.
Yeah, and for the CROs and the industry folks with AD and PD job, like process development, I feel like having starting with the basic knowledge. Like everybody needs to start at the same spot. So you need to have a training platform saying, hey, this is what we're going to do, all of these things. So having those things is set in place will be fantastic. And even before, I'm sure they have something like this in any of the teams you are speaking with. But if they don't, I would recommend, or ideally, what I would like to see is having a powwow hour every week or so, whenever you're creating a panel. Say, hey, this is what we're gonna do. This is what I'm thinking. What non-judgmental, like, because everybody, even if you are someone who has been working with photometry for like decades and decades, you will have questions. Right. So it should be a non-judgmental space where you could come in here and say, hey, this is what I'm going to do. What do you think about it? Just have this kind of a conversation, open forum, so that people can discuss it and then catch things that are gonna be something that could have been prevented even before you started, right? So I feel like those kind of resources in place and having open conversations will definitely help, especially in industry, because we kind of try to get siloed because you're in 80, you're in PD, you're doing this, you're doing that, because we get kind of siloed and we all have this own things. But when we just open it up and we speak, I feel like it might just be much more productive and economical also at the same time, because they're not cheap,
Absolutely.
man. High parameter reagents are not cheap.
Yeah, just the reagents to run just a few samples alone, it's gonna cost you. So, yeah, might as well do it right. Yeah, again,
Yeah,
not to
and
make it a sales pitch, but you're absolutely right. Like, that's why I think all 3 of us do what we do is for exactly this purpose, like, help people
yeah,
use
just
the
bring us in,
technology
right?
better.
Yeah,
Yeah,
like bring Ionic in, and then you don't need, like, just have a consultation. Say, hey, this is good practice. This is how you do it, so that you could understand what needs to go into it. Same thing goes with FlouroFinder too, because we may actually, all you need is just an account with FlouroFinder. Same thing goes with Christian's Supernova as well, right? You
right?
are able to just hop on the site, see what's in the site, understand what is available, so that you can make your choices and be informed, right? Instead of saying, hey, okay, fine, I'm going to willy-nilly do something. You can have these resources on hand, come up with a game plan, and then do it. At the ultimately, I feel like you will definitely save more money and have more confidence in your data rather than just running up with, okay, fine, I'm going to do it. So I feel,
Exactly.
yeah,
Would
like consultation is going to save you hundreds and thousands of dollars at the end of the day.
yeah, at the end of the day, I agree. It's the part that I think a very quick way to save money and get reproducible data, I would say titrations. You're going to get
Oh,
much better data. Manufacturers, antibody
Don't trust
manufacturers
the
always,
manufacturer dilution.
I
yeah,
wouldn't say don't trust it, right? With a
It'll
grain
work.
of,
It'll work usually, right?
it usually will work, but most of the time it's more than what you need. And more reagent is not always the right thing to do,
what's going to
especially
cause
when you're
spread?
doing, yeah, especially when you're doing a high parameter panel, you have to optimize it. You can't just say, hey, I'm going to do 2.5 microliters of everything. Like, nah, that's not how it works. So I totally agree, Christian. Antibody titration is a must and must and must. And especially titrating within every lot, right? It's not once and done.
I don't know if I would agree with that, Padmini. I think I
How
would
about
say,
tandem dice? Do you think you could just leave tandem dice as such?
oh,
Every,
well,
every
I have a personal animosity with tandem dice.
sorry.
I usually recommend people to stay away from them because they're so finicky and prone to degradation, bleaching, etc. So I say unless you have to use them, and sometimes you do, right? And it is what it is. But I usually tell people stay away from tandems and stick to primaries. But if you have to, then you do.
But in this day and age, with all these high parameter panels, you are unable to stay away from tandems. But the important thing, what I was trying to say is with the tandems, you definitely have to titrate it. You don't have to do the 8-point titration. You definitely have to do at least a 2 or 3 point titration from the
Right.
original just to make sure that you're within the ballpark, right?
Yes.
So
I
that's what
think
I'm
when
saying.
Yeah, I think when you qualify it that, I mean, when you put it that way, I agree. I just disagree of doing a full titration. I think
You
when
don't
you're
have
getting a
to.
new lot, you just do a lot bridging. Like you said,
Right.
maybe 3 to 4 points to make sure that everything is where it should be, and you move on. And you
What
don't
I've
have to
done
do so
before,
many replicates, I don't
sorry.
think.
No, I would agree. Yeah. I mean, what I've done and designed assay testing like this for like clinical trials. Granted, this is in Cytometry, so we don't have to worry about tandem guys, and metals are much more stable. But the bridging is basically like an additional time point in the interassay precision testing. So like we do our testing, we use the same reagent lot, new lot, same reagent titration level, and then just if it falls within the exceptions criteria thresholds, then we consider that good to go. Because most of it's like population readouts, right? Like it's not necessarily expression intensity. So it's much easier just to do it that way. Like if you're seeing the same populations, your reagents are just as good. Consider them equivalent.
And I would say with your titration, you'll also then be able to define your SIs. And well, for those that may not be familiar, that's the stain index. And that's going to tell you whether or not you have the right value for your titrations. Yesterday, on that point, I had a meeting with Rui Gardner, the director for
I love
MSKCC
Rui.
Flow Court, right? Rui's amazing. And he shared with me that in his core, they developed an algorithm that you put the FCS file inside a folder, and then that folder has an algorithm associated with it where it will scrub the FCS file to determine the SI of your data. I mean,
Mm.
provided that you've done the titrations, presumably. But then it will calculate the SI for you, and it will also do the if I remember correctly, and I could be mistaken, but it will also calculate your titrations. So that's like a really cool feature because like I posted on LinkedIn. I think I posted on LinkedIn, but if not, it's definitely in the LMS spreadsheet to auto calculate the SI and titrations, but you still need to enter the results manually. And what it seems that Rui did is that it just automatically does it for you by scrubbing the FCS file for the data or FCS files, right? Because you're going to have multiple titrations. But I thought that was really cool. I don't know. What do you guys think about
I wonder,
that feature?
I love it. I wonder if it's related to this. I'm just sharing my screen real quick. This is an omic. They just released this maybe like 2 weeks ago. I put a post on LinkedIn about it, but this does exactly what you're saying too. Basically, you can set up like your gating, and then you can create they have a stain separation index task that you
Mhm.
can essentially put your positive and negative gates. So it kind of works similar to their compensation wizard that they have.
Eric,
Uh,
I'm so sorry. I don't know. Can you tell for the audience what software this is?
oh, sorry. Yeah, this is
Okay.
Omic.ai. So it's super. I like Omic because it keeps putting out all these kind of cool features. And when you were talking about Rui's thing, it sounded very similar to this, where you basically set your gates on your titration data. So like here we have multi-point titration and can set our gates accordingly, and then it will automatically calculate your statistics for like your positive and negative medians, the standard deviation, stain index, separation index, and signal to noise ratio. So then you can
Mhm.
go in and you can generate automatically this figure, which gives you each dilution's histogram that you can see kind of the distribution of where the gates land, gives you your traditional kind of titration curve dilution plot, and then also gives you stain index, separation index, and signal to noise ratio. So it's super cool. I think also, like, I don't know how you guys approach it, but this leads to a question for you. Like, I find that some of these metrics, it's hard to really set a definitive point at which everything is good on these. You know, like you can have something that has really great staining index, but
Mhm.
poor signal to noise ratio. Like if we look here at the lower concentrations, we have the highest stain index, but really it's also one of the lowest, if not the lowest, signal to noise ratio. So like, how do you guys actually go about choosing the proper dilution level? Curious on that.
Padmini, do you want to take that on as our resident expert for, or representing FlouroFinder as the resident expert for panel design?
So for me, signal to noise ratio is really important, right? So like that's something that you need to look into, and also the separation is important as well. So for me, I would look for the one that gives me the better separation, but still has a lower signal to noise ratio, so that whenever you have, because this is just single stain, when you have other things that is piled on top, the signal to noise ratio is really, really important. So if you have multiple factors coming in, and that's going to increase the ratio, making your separation really lower, right? So I want to, ideally, if you go back to that picture, I'm feeling like the second point would have been the better choice in that space, because you might correct me. It's been a while since I have done it, but I feel like the dilution of 1 to 100 or 1 to 200 would be better for me to use, even though 50 has a better resolution of the population, but the signal to noise ratio is better in the 100 or the 200.
Mhm.
So I would recommend using 100 or 200. I will start with the 200, and if I don't get a great separation, I'll go to 100. But that's what I will look into, because signal to noise ratio is really, really important, because that's going to get exponentially increased as you add reagents to this thing. So that's
Right.
my thing. But another thing, what you can do is maybe actually select a different reagent for this one. It would have been a better choice. So there's a whole lot of things that we can do. But within this constraint, I feel like I would go with the ones with the better resolution, but signal to noise is also definitely important to look at. What do you think, guys?
Well, I would say to reduce your signal to noise ratio,
Yeah.
I would take a step back from the actual experiment or deciding the points, and I would look more at the actual technology. So not all detectors are created equally. Some
Okay.
detectors have an increased noise to signal or signal to noise ratio associated with it. And so when I'm working on to do a panel design, I first want to know what instrument the person has and what's the configuration. Because then I could start assigning floors or choosing or saying avoid this floor in an option. Like when I use, for instance, if I use FlouroFinder and it gives me a choices, I sometimes will reject and choose a different floor if I know for that particular instrument that detector has a lot of noise. Then I definitely need to look for a far more stable antibody. So
Absolutely.
that's part of when I start building an Ishikawa diagram when I'm doing my panel design. So assessing the risks. So before we even get to that stage, I'm like, is this going to be a good pairing of the antibody to that detector? And it's interesting that in spectral flow, they're starting to do this in the unmixing, right? So if you look at, I believe Cytec does this, that when they're doing unmixing, they do optimize unmixing, meaning I'm sorry, weighted up unmixing. And that meaning that they give priorities to different detectors based on on the known signal to noise ratio of those detectors. So I think that's where I would start before I even go any further. Afterwards, then I start looking at the antibody because I know that the more antibody I add, the higher my signal to noise ratio because of spread and the way it interacts with other antibodies, like Padmini said. So then at that point, I may want to reconsider using something else that has probably a different excitation amount and then a different emission, or just entirely change that antibody and put something else if that's possible. Sometimes that's not the case, and you just need to make do with what you got. And I think that's one thing I do like to tell when I'm doing consulting for people, that I tell them, you don't need to have a perfect panel because perfection is the enemy of efficiency, and you could just get stuck in that. I mean, I've gone through that road before where I go down a rabbit hole and just keep saying to make this
Optimize,
better.
yeah.
Yes. And your returns are diminishing every single time. So at that point, it's like, am I really going to get better data, or is it going to increase the quality of my data in an observable manner? If it doesn't, then I'm like, okay, then I need to stop and use what I have.
That's a great point. At least, yeah, you don't have to have a perfect panel. I would agree. At least if you go through the steps like we've kind of outlined here of planning everything out, though, you can at least identify those issue points or potentially break points or things that might come up when you actually run the experiment. That way, you know and can qualify. Like, okay, I know I'm going to have some spill in these channels. I can at least account for it if I know that's going to happen in advance. And then you can have, like you said, design the panel in such a way that, you might have some spill between CD4 and CD8. Put them next to each other. It's fine because they're not going to really be co-expressed in most applications unless you're looking at assignments. But like, yeah, designing it so it's not perfect, but at least the issue points you are well controlled for is very helpful and allows you to go. Yeah.
And I will say, FlouroFinder, maybe Padmini could mention this a little bit, and I'm so sorry I have to go because I have a meeting right now at 11. But one feature that I really like about FlouroFinder is the co-expression feature, where it helps you not put antibodies that are going to be co-expressed. That sometimes you forget to think about that when you're doing your panel design. So I really appreciate that you brought that up, Eric. Maybe Padmini, if you could elaborate on that. I'm so sorry that I won't be able to stick around for the explanation or the finishing of the podcast, but I do have a meeting that I have to go to. So I'm so sorry for this, but I have to step away.
Having the information about the co-expression group and also knowing the other reagents that can go in the same detector is really, really critical. And our platform will be able to do that for you. Like entire talk that we had today is thinking about everything that goes into a panel design, like not just panel design, but moving forward with that. So the main reason why even I didn't do that when I was designing panels and running is because of the fragmented resources, and having everything in one spot is going to be great to be able to do the due diligence and not like spend hours and hours beyond on that. So that's the main reason. That's the Achilles heel that keeps people from doing it, right? I feel like that's the so I feel when with the FlouroFinder, it's not a plug. It's just me as a researcher have utilized FlouroFinder. So I'm just giving my piece of knowledge. So FlouroFinder actually brings everything together in one spot. So even when you hop on the site, you have different tools like the antibody tool where you could just go and find any antibodies that you want, or the dye detector tool that will give you information about every dye out there. So it gives you information that you would require to understand your panel design, right? And even when you are on the panel design platform itself, you have the co-expression group thing that will tell you, hey, you have this in here. Even if you're going to do a manual panel build, it will tell you, please do not pick this reagent because it is going to have intensity mismatch or it is going to interfere with that co-expression group. So you get all those information beforehand so that you will not do anything that is wrong.
That's super helpful. It's such a nice tool to have everything in place in one spot, like you said, so people don't have to search
Yeah,
all
like
over and find everything. It's
yeah,
super
and it is
helpful.
free too. Like almost all the resources that we have on our platform is free to use. There are things that you can get a subscription for, like the automated panel builder. But even without automated panel builder, if you understand how to build a panel and you have all the information at one point, you should be able to build a panel without an automation, right? So that's what FlouroFinder. You could do that on FlouroFinder for sure. So I just opened our panel builder. I don't know whether I'm sharing.
Yeah,
I am
I
sharing.
can see it.
Okay, cool. So even like I just uploaded a panel here just to show you how it will look when you have already put in your data. So all these are antigens that I have already put in there. But what it gives you is actually the antigen density, because that's it's important, right? You need to understand your antigen density and also the all the other reagents that are available in the market. So it is actually important thing is to pair the antigen density to the intensity. So it will show you only those you can pair. Like lower density you can pair with high intensity, right? And if this is the high density, it will show you products that are lower in intensity. That's something that
That's
people
awesome.
forget sometimes. The brightness is missed. So you go and put your dimmest reagent in a, sorry, less abundant in a dim reagent. That's major no-no. And people
Yeah,
sometimes do
bad.
that, right? And other thing that you can, that this is what Christian was talking about, the co-expression group, where you can come and put in a co-expression group, and we do not populate it by ourselves. You are forced to populate it by yourself. Say, if you add a group, you are selecting the ones that you know that is going to be in that co-expression group, and you can also go into the cell types tool to get that going as well. But all these tools are holding your hand and saying, hey, did you do this? Did you do this? Did you do this? Right? These are things that people might forget sometimes and choose the wrong reagent and run it. And then have the issues with the data antibody or data spread and all those things. So we are trying to give you all the information beforehand. So you will think about all these things when you're selecting a panel. Other things what Christian was talking about is over here. Let's go ahead and do this one. Let's do this. Sorry. This is just a backend thing. We don't want to see
Oh,
that.
no, all good. Yeah, this is
But
such a helpful tool for people, and it's amazing that so much of it is for free. So, like,
it is for
people should
free.
at
Like
least
all
be
these,
using
everybody
this
should be using this. It's not
if you're
just because I'm
designing
with
something.
FlouroFinder. It's just we are trying to make flow cytometry research much more easier and approachable, because sometimes people don't think about it because they feel it's too much. Because yeah, it is too much because there is a whole lot of thing that goes into
There's
it.
so much, yeah.
There is so much. Like I have felt overwhelmed, right? But when you have a tool that does the thinking for you and also shows you what it is doing, that is what I feel like it's really great, right? So what you are looking at here is all the detectors under that laser line, and you have all the reagents that can go in each APD. So if you just click on that, it's going to tell you, hey, all of only if my internet is working properly. But anyways, if you see here, what it's telling you is in B6, you can have this reagent and this reagent. So it's giving you all the reagents that can be in each thing. So you can select the ones that you want to. But as Christian was talking to you about, hey, you don't have to have a perfect panel, but if you're able to switch between reagents that you have, yeah, go ahead and do that kind of thing, right? So, but anyways,
Right.
it's here. Go ahead and look at it. But its important thing is to understand that you cannot always start with a perfect panel, but you can start with a panel that is nearer to that, and then utilize all the resources that are available. Just definitely open up and talk. Or if you have a question, reach out to any of your peers or something, and then be confident in what you're doing rather than just doing something and then trying to bullshit your way out of it. That's not the right way.
Right,
Okay.
100%. The data will never lie. It'll tell you. It'll show if you're bullshitting or not.
Yeah, right. And
Always.
like That's the favorite part of mine. Like even when I was in the full cytometry core, run the instrument, running on instrument, creating that panel and all those things are fun. But the funnest part is when you're analyzing the data, right? So I used to love analyzing data, and when I was talking to you, I was like, oh my god, I want to get my hands on Omic and do some data analysis. Like it's I feel like that's a culmination of everything that you have done. It's like a realization of what you have done thus far. So it's a really great experience to do analysis, I feel.
Exactly. Yeah, when you design the panel, it turns out well, you look at the data, it's nice and clean, and then, yeah, like you said earlier, I really appreciate the, like, story of the data. That's what I try and say, like, what's the
Uh-huh.
data is trying to tell a story. Our job when we analyze it is to learn what the data is trying to tell us, really. So it is very cool to see it come to life.
And the thing is, you can actually tell what went wrong where with just from the
Right,
data. If you did it right, if you do the analysis properly and you know what you are doing, you can tell, hey, looks like there was a bubble when this ran through. I can tell you from the
right.
lag I see. So it's a understanding of how you got the data and you're able to say everything from that. So it's really I enjoy data analysis for sure.
Yeah, the data doesn't lie.
Data never lies. Yes. So enlighten me a little bit. I know in Ionic you do a little bit of consultation, right? So, and like,
Mhm.
tell me how rigor and reproducibility is like, how do you talk about rigor and reproducibility from comes from Ionic?
For sure. Well, I mean, like I said, it starts really I try and get with people before they actually do anything. A lot of times people come when they already have data, and so it becomes more, okay, what can we get from this data? How can we
Mm-hmm.
kind of
Uh-huh.
make the most of it? And then obviously document everything in the analysis. That's why OMIC is very nice because it's all auditable. You can see exactly what was done to the data, and then what's put out. But then really it comes upstream of like helping people plan their experiments, design their assays, and then building in the controls to really make sure that things are running properly. So I always try and, especially with like Cytoff, which is something big that I do, and then also with Flow, is having a reference control in all your experiments. Some kind of thing that you can reference. Like cells are great. They're really helpful for that. Some kind of even just a technical healthy donor, like Very cells, something like that. I really like them for the ease of use of just having, okay, is this panel changing in how it's functioning over time? Hopefully not. If you design it well, it shouldn't change all that much, but it at least allows you to catch that and maybe control for it. And that really is where the reproducibility and the rigor comes in, is the planning. Like it's kind of a broken record at this point for this
Uh-huh.
what
Uh-huh.
we've been talking about, but it really is true. Like I've seen in my experience, if you do that, you don't really need to do a lot of batch correction. Like I want to
Yep,
avoid manipulating the data as much as possible. And the best way to do that is to plan it out properly. And it
definitely.
holds up. It holds up. Like obviously there's some batch out correction algorithms that work fairly well, but
Uh-huh.
they always make me nervous because I think I'm also, well, not I think, I know I'm also going to be getting rid of some biological signal. And so I don't want to do that, you know?
Yeah, it's
So
like
that's kind of
working.
how I approach it.
Yeah, it's like it feel like it's similar to what we are doing with ChatGPT and Cloud AI, right? So we are trying to put things in there, and I know for a fact that it's not giving me what everything that I need if I had sat through it and read through a paper, right? So it's
Right.
I guess it's always you take it with a grain of salt when you're doing all these batch corrections because you know that you are sacrificing some of your data for sure, right? And if you have control for those things even before you started, definitely would have been better. But Ionic, I feel like you'll be able to handhold them starting from the start of the project and then actually walk them through and then be able to help them at the end of the project when they are able to analyze the data for them. So that's a great thing to do.
Exactly, exactly.
That's
That's the
awesome.
goal, just to help people use it better. Like these tools, I think that's the common theme of all of us here. Like these high parameter cytometry tools, whichever one you're using, they're so powerful and can really tell you a lot and help figure out a lot of interesting questions. So it's, but like we said, they're complicated and it's easy to mess up. Sometimes it's hard to do well, especially if you're just starting out. Once you get into it, you realize like as long as you know what you're doing, it's really not that complicated, but it's easy to mess up, and so it's good to have some help to do it properly and get the most
Yeah,
out of it.
I definitely and I totally agree with that because especially when it comes to high parameter flow cytometry, be it cytoph or fluorescence, right? I feel like anybody, not just CROs or the industry, like anybody who's working with those, they got the science. Like you know what you're doing about like the cell types and everything. You got the science, but they always definitely could use some help regarding the technology, and that's where we all come in. Like you or
Right.
Christian or me, when I say me, FlouroFinder, we all come here to help them do better science. And I feel like at the end of the day, whatever you're shelling out in the earlier part of your planning is going to be given multifold when it comes to you being able to release a product that you are very, very confident about. Right. Right. I feel like it's a no-brainer for sure. And if I were in industry, I would definitely have any kind of help I can have because at the end of the day, I love where Christian talked about in his paper. He was talking about having it as a sequential approach. It's not just one or the other. It's like sequentially starting from one to ending with the process development and having a validated assay that's been released. So I feel better preparation early on is going to anyways definitely help with better returns in your
Exactly.
time, right? It's
Yeah.
not just money, it's time too. So I feel and confidence in
And
what I'm
sa
yeah.
yeah, confidence, you wanna be sure of what you're saying. Yeah, that's, uh, that's definitely the big thing, you
Mm-hmm.
know, and yeah, that's why you're talking about reproducibility and rigor too, something that we do a lot with our analyses that especially in the more discovery type of space where we're like finding new things, is
Mm-hmm.
analyzing things with different algorithms, the same data over and over again with different sorts of perspectives. And if we see the same signature in different angles, then we're pretty confident that we're getting a real
Yeah,
result,
right.
you know, rather than something spurious.
Yeah, and whenever you're doing these kind of discovery studies, it's not something that is a huge population anyways. It's going to be like 0.001% of your population, and you need to be really, really confident about the SOP that you use to get over there, right? So you should be
Right.
I feel like it's you are shooting yourself in the foot if you don't start with a better panel design or a plan to how you're going to do analysis and stuff at the end of the day. So yeah. Rigor and
Exactly.
reproducibility is my two favorite R words.
I love it.
Yes.
Yeah, it all boils down to plan, plan, plan. That's
Yes,
the big key.
definitely. Definitely.
Yeah,
Awesome.
amazing. Well, thank you so much, Padmini. Do you have any parting words for people or a guest? I mean, we'll put a link to FlouroFinder up when we post this, and hopefully people will go and use it.
Mhm. Because all these are free resources, and I do not know about your consultation either. Like they can reach out to you and consult and have an hour with you just to understand. Like because it's a
Oh
no
yeah,
brainer. It's a no brainer. Then and I don't think there should be any excuse to be not able to do better science when all the resources are available, right? And I'm glad I got to talk to you guys and be able to tell you, hey, what's available out there, and also learn more about Ionic and the supernova as well. So it's great. So thank you for having me for sure.
likewise. Yeah,
Yeah.
thank you for joining us and showing us FlouroFinder, and yeah, we'd love to have you back on sometime.
Yes, and hopefully I'll get my Rajapaliyam too.
Yeah,
The dog.
I love it. I
Yes,
love
yes.
it. All
Thank
right,
you.
Padmini, have a good rest of your day. Thank
You
you.
too. Appreciate it, Eric. Have a good one.
Bye.